When we think about cell and gene therapy scales and industrialization, one question that needs to be answered is what scale is needed to match the therapy demand. This is very dependent on the therapy targeted, because as you see here, the scale that is needed is a combination of the number of patients per year. This will be influenced by the incidence or prevalence population, the therapy access reimbursement of the country or the region, and the market share. This is factored by the patient dose. This is very, very variable depending on the therapies that are targeted. As you can see on the table on the top right, it can very easily range from 10 to the 11 until 10 to the 14 or even more. And another factor that comes into consideration when thinking about the scale is the number of manufacturing rounds per year. Because if you do too few, so you choose a scale that is too big, your operators will lose training, they might do mistakes, it's also an inefficient use of capital, and if it's too many, you have a higher risk on the supply chain. So there's an equilibrium to be found there. These are some examples of calculations depending on the indications, the number of patients that are targeted by these indications, and the doses. And you can see here that it can very easily range from 10 to the 13 to 10 to the 18 viruses per year. So it means that really this needs to be taken into account to decide what scale should be needed. So when thinking about the scale, one question also very often arises with our customers is what platform should I choose? Should I go adherent or suspension regarding the cells? This is something that is to be tackled as soon as possible in the process. And it really depends on a lot of factors, whether your cells are already in suspension, whether they're still adherent and you would need to adapt them to suspension if you wanted to go suspension. What is your final target scale? So because, for example, if it's very, very large scales or 2,000 liters or more, then single-use suspension would be probably more appropriate than adherent. But there are a lot of things that need to be taken into account, and we can help you with that decision. So within our process development services, we, whether it is an adherent or suspension process, we usually go through several steps, such as first, as I explained earlier, the tech transfer, then the proof of concept, and then the process development and scalability in the bioreactor, where we can look at a lot of different parameters, such as seeding densities, transfection strategies, etc. We normally then reproduce the optimal conditions, and then we might scale up if needed. Ideally, we do 2 scale-up runs, and then we would transfer back to manufacturing. So I will now present you 2 case studies around suspension cell cultures. So the first is an AAV suspension scale-up production case study. So this was done for a customer where we evaluated the Allegro STR bioreactor 500 liter for AAV production by HEC293 transient cells. So we got the process in shake flasks. The goal was to transfer it to an STR 50-liter bioreactor for the seed train, and then use the seed train in this 50-liter bioreactor to seed our SCR 500-liter bioreactor for the AAV production step. This slide shows the comparison of growth of the HEC293 cells in the STR50 bioreactor compared with the shake flask. You can see on the graph on the right that we have a comparable VCD and viability obtained in the Allegro STR 50 liter compared with the shake flask. On the next slide, we can see the comparison of the AAV productivity by HEC293 cells in suspension using different STR bioreactor scales. So we have the productivity in the Allegro STR 50 liter and the productivity in the Allegro STR 500 liter. And we have also comparability to two other vendor bioreactors, 200 and 500 liters. And you can see that within our STR range, we have a very good tighter scalability from 50 to 500 liters. And we can also, and this process can be easily reproduced and transferred from competitor bioreactors. So the performance is comparable. The next case study is around AAV scale-up production with suspension stable cells. So these cells are a CAP cell line from CEVEC, which is an immortalized human amniocyte. They are stably transfected with genes for recombinant AAV production, and the production is inducible with doxycycline. The customer transferred us a 10-liter STR process, and the goal was for us to scale up this process to 50 and 200-liter bioreactor. And the scaling strategy applied was our standard one, which is maintaining constant power input and maintaining superficial gas velocity. So to do that, we have technical tools that we created that allow us to input the customers or the starting bioreactor characteristics. Here we assume this was the 10-liter BioBlue of Eppendorf. So we input the parameters, and the tool automatically gives us the scale-up parameters in our different bioreactors. So we can therefore also enter the agitator and airflow rate speeds of the bench scale STR in this tool, and then determine the PV and the superficial velocity for the constants for the scale-up that we need to apply. And so the model will back calculate the RPM we need to apply in our scale-up bioreactor and the liter per minutes that we need to flow. Some additional considerations that might be needed to be taken into account are other variables such as the Kolmogorov length or the tip speed, but these are normally accessories, and we tend to have very good results with our approach around power input and gas velocity. So with this approach, this is a graph that illustrates the cell density, the viable cells in millions per ml, and the viability in dashed lines of the 10-liter benchtop bioreactor, which is in gray, and our STR50 and STR200. And you can see that overall we have very good scalability between the STR50 and the STR200, and we even reach higher cell densities and better viability than in the 10-liter benchtop bioreactor. Regarding titer of the AAV, here you have the VGs per ml of the harvest. And you can see that the comparison between the 10-liter bench scale and the 50-liter or the 200-liter replicates really are comparable. So we have a very good scalability within the STR range, and also from another bioreactor, such as this 10-liter benchtop bioreactor. So in conclusion, these studies show that AAV processes from customers transferred to us have been successfully scaled up into our PALL STR bioreactors. The AAV process scalability in PALL STR bioreactors is independent from the scale-up strategy applied, whether it's tip speed or power input, even if we tend to prefer power input. And our PALL Accelerator Process Development services will assist customers in scaling up their process to meet clinical trials and market demands. And with that, thank you for listening. Thank you.